We are live.
Okay, great.
I was like worried that there was going to be some sort of miscommunication with the GMT versus summer times or daylight savings times.
Yes, yes, it worked out.
Yeah,
Hello,
worked out.
Boris. Good morning.
Hello, George. Hello,
Uh,
Fabian.
yeah.
Morning.
Hey, Boris.
How are you?
I'm fine.
Good, good, good.
Good,
Early
yeah.
morning for you.
Yeah, it's early, but yeah, it's already daylight.
Okay. Good morning, Poojitha.
Hi, George. Good morning.
Good morning. How are you?
I'm doing well. How are you?
Thank you. We're doing well. Thank you. Yeah.
Good morning, Fabian, Boris. Boris, very early for you. Sorry.
Yeah, it's fine. It's okay. So how long is the plan is to in the entire session will be 90 minutes?
Yes, as needed.
Mm-hmm.
Yeah. By the way, we are live, right? It's the recording is underway.
Okay.
Yeah.
Awesome.
All right, good morning, ladies and gentlemen. I guess we'll get started. It's 10 o'clock Eastern Standard, or 2 p.m. Greenwich Mean Time. My name is George Akwa-Mensah, Convention for Biomedical Research, Ghana. This morning, we are privileged to have a team of experts to walk us through how we get to access and harness cancer data in the cloud for our research. Without much ado, I will attempt an introduction. I mean, you can fill in the gaps because I didn't really get all the information I should need. But we have Dr. Boris Aguilar from the Institute for Systems Biology and the General Dynamics Information Technology. With him is Dr. Fabian Seidl. It's also from the General Dynamics Information Technology, Senior Bioinformatics Engineer. Also, we have Dr. Poojitha Gundluru, also from GDIT. So without further ado, I will hand over the proceedings to the team. Thank you so much.
Yeah, hi. So I'm Fabian. It's nice to be giving a talk here. So it's a real honor.
Before go, just a little housekeeping. Please feel free to post your questions in the Q&A or in the chat, and we'll address them at the appropriate times. Thank you.
Yeah, so I'm Fabian. I'm going to be giving the introduction to sort of both, what is bioinformatics data types? What are the bioinformatics data types? How do they apply to cancer research? And then sort of a background of the CRDC and sort of the cloud initiatives that were started recently. So I'm going to be doing a little bit introductory here. I'm not sure what everyone's background is. So if you already know some of this information, just bear with me. It'll be brief. Okay, so this is the agenda that we're hoping to share with you today. First, there's going to be an introduction. I'm going to be talking about, derived bioinformatics data, cancer initiatives, and then what I like to term sort of the problem, which is, why are we here? Why do we get funding from the NCI? What is it that we're hoping to accomplish? And then we're going to go over the actual sort of cancer data, the sources where that cancer data comes from, which is sort of an introduction to the NCI and the CRDC, and then the data commons. And I'll go into what the data commons are in a little bit more detail. And I'm going to be navigating through their portals and sort of showcasing how to use these resources, or how to get to the data that you might be interested in within these resources. So yeah, the study of cancer genomics. Cancer genomics is an intersection of a lot of the advanced sort of, second generation sequencing technologies that came to the forefront, about 10 years ago. So when they really started kicking off, when sequencing cost dropped to affordable for for a common lab. And then, the computer science that's required to interpret these data, to get them into sort of a derived format, meaning, a quantitative final output that you can actually start running statistical tests on. And, to really get at studying cancer, we need a lot of data because it's a very complicated disease. It's a very heterogeneous disease. And, any amount of extra data can be the difference between sort of achieving an insight and not achieving an insight. And so there's really a wide, scope of bioinformatic techniques that are available these days, and a lot of different, data types that when combined together can really drive insight into cancer. And so this is a little example where it's mainly using sequencing data to achieve germline mutations and somatic mutations by doing DNA sequencing and then aligning it to the reference genome, and then getting out those mutations, and then combining it with the sort of clinical and annotation data such as, what are the environmental factors, what viruses is, they're inherited, is there a history of cancer in the family, et cetera, et cetera. And so I want to break down sort of the major, data types that I'll be talking about later. So the first one that I've already touched on before, high throughput short read sequencing has really just ballooned or exploded in use across the biology community in general, but specifically in cancer, it's been heavily used, and there's a lot of data out there. And so the way that this works is you take your tissue sample, you lyse the cells, you extract the DNA from it, and then the general methodology is you shear it into small reads, which are then loaded onto these Illumina machines. Illumina is really the main company and the main platform that's used for short read sequencing. And then these short reads, generally there's a PCR step involved so that you have an amplification of the genome, and you're also combining multiple cells from a large tissue sample. And so there's many different copies of the genome in there, some of them biological copies and some of them technical PCR copies. And then you get these, between 100 and 300, 500 base pair length short read sequences. You align them to your reference genome, and then you can do several different analyses from this point. You can, perform genome assemblies, de novo genome assemblies, by looking at the overlap between them and building out scaffolds. You can run this analysis not just on DNA, but you can convert RNA into cDNA and then run these libraries. And so you can do an RNA expression analysis from them. You can look at the differences between your short reads and the reference sequence, and then use that to call mutation absence and presence, both these single nucleotide polymorphisms, as well as sometimes longer indels, so insertions or deletions. And you can look at copy number variation. So by looking at the read depth of any given part of the genome, you can ascertain whether or not that region has been enriched compared to what is expected or depleted. So, in this case, you can see that this region is about double what this region is. And so one could infer that maybe there's a 2x amplification here, or there's a loss of heterozygosity or something along those lines in this region, or a loss of this gene on one chromosome or something. And then the last one that is a little bit more of a specialized protocol, but uses the same libraries as far as I know, you can get methylation status. So is that region methylated or not, which can have implications on transcription of genes in that region. The second major data type is proteomics. And so there's been recent advancement in this in higher throughput proteomics. And so that this is a measurement of the abundance of a protein in a given tissue sample. And, it's all relative, but I think they do up to hundreds of genes these days, which is a little bit below the level of DNA sequencing still, but it's a pretty exciting field in general. And the central dogma of biology is, a gene is transcribed and then translated in the cell. And so one would think that one can get a lot of the information of protein expression directly from the RNA expression, but oftentimes there is a disconnect there where certain RNAs are expressed transiently, but the proteins that are formed from that end up sticking around in the cell for a very long period of time. And so that can there's a lot of extra value to be gained from having the protein expression values directly as well. And then the last sort of major data type that is present in the CRDC is imaging data. And so not just the slides, but there's also a big push to get more quantitative sort of cancer phenotype data out from these images. And so, machine learning is a burgeoning field as well. And it's really the promise of being able to automatically annotate the large amount of data that's available, because the manual annotation that's been the standard up to now is very work intensive and takes a very long time. And then there's a lot of inconsistency, sort of when multiple different people are annotating the same tumor or the same images, they don't always necessarily agree as much as we would like. And so having a standardized, effective, and specifically fast method of annotating tumor images is sort of the holy grail. And then, the ideal is to get out information that one can actually run statistics on. So what is the volume of that tumor? Sort of what are the major shape characteristics? Like, for example, how long are the axes? What is the texture of the tumor? That's something that could be of interest and could have sort of implications on the growth, etc., of the tumor. So things that we can kind of phenotypes that we can take from the images and then link back to, you know, RNA expression or something to really try to get an understanding of biologically and mechanistically what's happening in this tumor and what does that imply for the patient and what treatments might be more or less effective. And being able to sort of identify these sort of things directly from the imaging data is one of the main things that I think clinicians are very interested in. You know, can we image a tumor non-invasively and then have some sort of idea of what treatments might be effective? And this is still in the infancy, sort of in its early stages, but I think there's been a lot of progress recently, and it's a very exciting field. So yeah, so this is a lot of data, a lot of different types of data, and there are a lot of initiatives out there, such as this main example, the TCGA, the Cancer Genome Atlas. You might have heard of it. It's a huge initiative, 11,000 patients, 33 different tumor types. And they went through and they generated these types of data for all of those patients and all of those different tumor types. And it ends up being an overwhelming amount of data. It's 2.5 petabytes. You know, it's getting to the point where it is so much data that an individual lab or an individual researcher really can't feasibly download it all onto their computer to sort of do these analyses. But the whole intent of these projects was to make this data available to researchers all over the world for free, and then to thereby sort of drive cancer discovery and cancer research. And so, you know, faced with this overwhelming amount of data, how do we best present this to researchers? How do we best make it available? And so there are many, you know, TCGA isn't even the only initiative. There are many more. So CPTAC is another one, which is the Clinical Proteomic Tumor Analysis Consortium, HCMI, which is the Human Cancer Model Initiative, and then Target is another huge one, which is therapeutically applicable research to generate treatments. And so, you know, not only is TCGA so large, but there are sort of initiatives that are nearly as large or as large that we also have data from. And then the additional problem on top of this is that they might have all used different methodologies for analyzing their data. So when you get back these short read sequences, you align them to a reference genome, and there are many different aligners out there, so different software tools that perform fundamentally the same function, but might have small differences in how they end up doing it. And so your final data output might be a little bit different. And those are all, you know, confounding factors that if you do end up wanting to combine data from these initiatives or, you know, compare data from these initiatives, you'll have to, as a researcher, work through and try to, you know, tease apart to get to the actual biology that you might be interested in. And so, you know, this is obviously kind of a tangled, difficult, thorny problem. And so one of the big initiatives that was started was the CCDH, which is the Center for Cancer Data Harmonization. And so the idea here is they will take all the data from these big projects, they will reanalyze them using the exact same methodology, using the exact same pipelines and the exact same programs to try to reduce that confounding factor of, you know, the analysis to the minimum and make these data as compatible as we sort of can. Of course, there are still considerations. You know, a different lab might perform the library preparation differently, but, you know, at the very minimum, try to sort of standardize as much as feasibly possible. And so this is showing one of their workflows. This is an alignment, I think, an RNA sequencing workflow where they use the same, you know, HT-seq and Fusion and Ariba, which are multiple different pipelines to sort of generate the final output. And all of these sort of workflows are available on their documentation page. And so if you as a researcher, if you're interested in doing this kind of work, or, you know, if you have done it in the past and you want to sort of standardize your workflows to use the same ones that are used at GDC to sort of make the submission process, et cetera, or the comparison process as simple as possible, that's all available to, you know, everyone, publicly available. And so once you have, this enormous set of standardized data, where do you store it? That's been sort of one of the main questions that's been facing cancer research. And so the CRDC was created. It's a cloud-based data science infrastructure. The idea is to provide secure access to all of these large and comprehensive and expanding collections of cancer research data, and then make them available to researchers. And also go through the sort of thorny problem of personally identifiable information, which needs to be, which can only be accessed if you have the right permissions, and kind of handle all of those logistics to still make it easy for researchers that want to use these data to access these data. And so I'm highlighting here, this is sort of the larger cancer research data commons, but the data commons specifically, the ones that I was mentioning earlier, the proteomic, genomic, and imaging data commons, which store the vast majority of these data, are kind of highlighted here. And I'm going to be going through them one by one in this next part of the presentation. So this is broadly the three data commons. They host and control access to different types of cancer data. It's, from their names, you can infer that the genomic data commons handles genomic data, proteomic is intended to handle proteomic, and the imaging is intended to handle the imaging data. There is a little bit of overlap. It's not, a perfect sort of broken down, there's not extreme barriers. For example, the genomic data commons does have some imaging data and some proteomics data, mainly because I think they were the first initiative to sort of arise. But the goal of the genomic data commons is to host genomic data. So yeah, this is sort of their mission statement. The NCI's genomic data commons provides the cancer research community with a unified repository and cancer knowledge base that enables data sharing across cancer genomic studies in support of precision medicine. And so they have, transfer tools and APIs and a portal. And here's the main data types that they host. So they have, the general read BAM files. They have the QC of them. They have, BAM files here, sorry, aligned reads. And then they have the quality control metrics for that. They have gene expression data. So this is more quantitative, the final output from these aligned reads. They have somatic mutations. They have the masked somatic mutations. So the difference here is, they have the germline as well. And then mRNA expression, structural variation, methylation that I touched on earlier. And they have all these different, in different data formats as well. Okay, so I'm going to be going and exploring the portal a little bit. So the first thing is we get a warning that this is, some information that is protected under the US Privacy Act because it contains some sensitive information. Most of the data available on this portal is completely open access. You can download it without having to have any specialized permissions. And so you can navigate this portal. They have a nice little interface here where you can select the different types of the body. If you're interested in a specific cancer type, you can directly subset it using this. And one thing I want to point out is their documentation is a little bit hidden up here in the GDC apps. You can click here and it'll take you to their documentation page where once again, they have the sort of workflows mapped out. They have an explanation of what each data field is for every data type, et cetera, et cetera. And they also have some tutorials for how to, download files, analyze data, et cetera. Going back to the portal, we can go through and explore this data set. So I'm going to click select exploration over here. And you can see that they have different subsets. You can subset the data by the primary site that you're interested in. That's similar to the homepage where you could click the different organs. You can subset it by a program. If you're specifically interested in exploring one program, you can easily click that here. You can go to projects, which are a subset of programs. Generally, these are also the primary site. They're broken up by primary site. For example, here, TCGA LUAD is lung cancer, for example. You can select a disease type. And then you can subset by primary tumor versus blood-derived versus solid tissue normal. You can also subset, up here, there's clinical data. You can access the clinical data directly here and subset by that. You can also jump to the mutation status. So you can look at whether or not they have, deleterious mutations as defined by this SIFT pipeline or the polyphen pipeline. And what you can also do is, select different file types. So if you look over here, they have multiple different files available for data category. So this is sequencing, this is expression, simple nucleotide variation. And then if you want to, download those data types, the mutation data is controlled, or some of the mutation data is controlled, but the expression data should not be. So here you can see it's open access. And then you can, for example, click this star counts, and then you can download the file here. Going back to the exploration, there's also this OncoGrid thing, which is, I think, really neat and a quick way of sort of highlighting the data that you're looking at. So this is, the mutations that are present in a sample. And you can kind of see that, the standard mutations. And if you subset by data types, I'm pretty sure this graphic will change. Yeah. So, TP53 is a very well-known cancer mutation, et cetera. So this is the GDC. This is the data that's available there, and there's a lot of sort of analysis tools that you can run directly in their interface as well. Okay, going back to the presentation. So the next one is the proteomics data commons. Similar mission statement to the GDC, but they're focused mainly on proteome data. So the CDRDC enables researchers to link proteomic data with other data sets, e.g. genomic and imaging data, and to submit, collect, analyze, store, and share data through the cancer data ecosystem. So this is the data types that they have available. They have, raw mass spectra. They have processed mass spectra. They have some RPPA, which is reverse phase protein analysis. Sorry, I don't know the last letter. And then they have the metadata and the quality metrics as well. So let me jump to their portal. You get the same warning, and their interface is very similar to the PDC or the GDC, I'm sorry. You can select the different organs the same way. And then we have their documentation is under more, but we can jump to explore. And here you can subset by primary sites similarly, program, disease type, analytical fraction. So this is something sort of unique, if you're interested in the acetylome or the glycoproteome, phosphoproteome, et cetera, you can jump directly to those. And then we also have, biospecimens, we have the clinical data, we have the files data directly accessible. And then you can, if you're focused on specific genes, you can browse by that as well. So those are different ways that you can sort of browse the same data. You can also enter a list of gene names that you're particularly interested in, and it'll tell you sort of the data available or the projects that have data with that gene in it. So multiple different ways of sort of slicing the same pie. Okay, and so the final one is the imaging data commons. And so, I kind of previewed that a little bit earlier with the images that I had. But generally, currently, they're mostly storing the raw images. There is an effort to sort of quantify those images, pull out the sort of metrics that cancer researchers might be interested in, and then those would be hosted in the same place. But it's still early stages, and so there's not too much available of that yet. So these are the data and the imaging data commons. There's clinical and preclinical imaging. They have the different types, radiology, digital pathology, multispectral microscopy images, the annotations for those images, how was it done, et cetera, all those sort of nitty-gritty details. And then the measurements of derived images, the annotated regions of interest, and expert assessments of those image findings. So let's jump to their portal as well. Okay, so they have the same sort of subsetting. And then they have a little bit different here. So they have the radiology or CT scan images. They have MR images. They have PET, and then they have slide microscopy. And so one of the cool things about the imaging data commons is you can directly access these images and you can directly view them in the interface. There's a little loading bar that I have to wait to finish. So this is stacks of the same CT scan. And we can sort of move through that CT scan. And you can even see here, there's some annotation information. So this green outline is the sort of the borders of the tumor as annotated. And so you can kind of see the extent of that. Jumping back to the portal. So 1 second. The documentation is up here as well for them. They have, they have a lot of information on sort of how to access these images on the DICOM format, which is the general imaging format that they use, which has, it's a lot of metadata as well, the DICOM. And then, some information on how to sort of browse the website. So definitely recommend that. So similar as the GDC and the PDC, we have the different body parts. You can subset by that. There's 78 of these. So, and then you can subset by that. There's a lot of these modalities, which are imaging specific, methodological characteristics, I guess. And then one of the things that is a little bit unique to the way the IDC handles it. So currently there are 0 cases selected. If you want to start exploring them, you have to, for example, select a collection name. And then the cases will pop up down here and it'll show you like number of studies, number of series. And then if you select a specific case, then it'll give you access to the actual data. And so, you can click directly on the view button from here. It'll take you back to the OHIF viewer and you can kind of, jump through the imaging data there. Okay. So those are the data commons. They host the different types of data that you might want to access. We definitely encourage you to go there and just sort of poke around, explore, and see sort of what you can glean from this information. There's a lot of it. It's worth sort of exploring and worth sort of getting an understanding of what's available there and how much. And so this is the one extra nice thing about these data commons is that some of this data overlaps. So there are 503 cases. This is somewhat old. I'm not sure if this is still accurate, but this is when I ran the analysis a while ago. But there are 500 or so cases that have data present in the GDC, the PDC, and the IDC. And so if you're interested in sort of a holistic analysis, there are definitely data sets. I think it's mainly TCGA and CPTAC that have this overlap, but there are, cases where there is imaging data, RNA expression data, mutation data, and some proteome data. And so that's, in my opinion, sort of the holy grail of cancer research in a lot of ways, being able to combine, all these different data types to sort of get a real insight to what's happening in those cancer tissues. But like I said, the data commons, it's intended to sort of standardize everything and bring it all together and host it, but it can still be an overwhelming amount of data. So for example, TCGA LUAD has 18,723 files, nearly 80 terabytes. And it has this across 17 different cancer initiatives. I think it's 18 now, actually. And even just TCGA, just transcriptome files is 33,279 files. And so if somebody wants to analyze these data, they might have to download all of those files to their computer and then concatenate them together into the same sort of data matrix, expression matrix, before they can even start running any statistics on it. And so that's an onerous process, obviously. And the downloads themselves could take a very long time. And so because of that, there are better solutions. There are alternatives to this. And that's sort of where we come in. That's sort of our motivation, is to try to make this whole thing easier and more manageable for researchers. And so at this point, I'm passing it over to Boris, who will tell you a little bit, specifically about cloud resources and how to sort of run these analyses in the cloud and what benefits that brings.
Thank you, Fabian. I will share my screen.
Okay. That sounds good.
And if you have questions, please let us know while I am setting up this presentation.
All right, Fabian, may I ask a quick question?
Of course.
While he sets up. So the 503 that are they cases or samples? Is it 503 patients for whom we have patients?
Yeah.
Okay.
Yeah, it's patients.
Okay. Yeah.
It might be a little bit lower than 500. Sometimes there are rare instances where one case will pop up in multiple different projects. You know, an individual that had 2 cancers or something. I think it's rare, but it might happen. So I don't want to say that that's exactly the right number.
Okay.
And it's also important to note that, you know, it's broken down. Those 500 patients don't all have the same cancer type. So, you know, once you start kind of digging into it, you might only have 20 or so cases for the cancer type that you're interested in that have all those data overlap, which is still a relatively small sample size, but you know, there's progress happening, hopefully.
Yes, it's a big resource. You know, we can look at the different types of data on the side. Thank you. Sorry to interrupt.
All right, thank you, Fabian. And my name is Boris Aguilar, and I work in the Institute for Systems Biology. And I will, in this part of the presentation, I will talk to you about ISB-CGC, which stands for the Institute for Systems Biology Cancer Gateway in the Cloud. And as ISB-CGC is part of the CRDC that Fabian showed it to you or mentioned before, and it's one of the 3 cloud resources that is part of this project. And it is funded by the National Cancer Institute of the US government. And in this project, it's a collaborative project between the Institute for Systems Biology in Seattle and the General Dynamics Information Technology. I think it's in Maryland. ISB-CGC is part of the CRDC, and the other 3 cloud resources are 7 Bridges and Broad Institute. That's a little bit of information about ISB-CGC. And here is our mission statement. ISB-CGC provides resources and store cancer data and provides compute resources in Google Cloud so you can perform cancer research. It provides interactive web tools. It also gives you access to the data through BigQuery tables. And it also provides workflows so that you can analyze the cancer data in the cloud. All the resources and the data that we provide is publicly available. So you can access all this data, and all the resources are also free. For example, we provide notebooks with Python scripts so that you can analyze the data. So those are free. So those are open, and you can access this through our webpage. Here is an overview of ISB-CGC. And for the cloud, we use Google Cloud, which offers several tools for data analytics. Specifically, we use cloud storage, in which you can save your files. You can put all your files in cloud storage, so then you will be able to access it through the cloud. And we also use BigQuery, which is a database of tables. And the point is that all these files are transformed into these tables so that you can use all these tables in the same format and do some analytics over your data. And Google Cloud also provides virtual machines which are computers that you can access in the cloud, and you can do analysis on this computer. So you can store your data, explore your data, and analyze your data in the cloud. And in ISB-CGC, we developed and we provide web applications, workflows, which are programs that you can run to analyze data, APIs, which are a set of functions that you can use. And I already mentioned notebooks in Python or R, so that you can extract data in the cloud, or you can explore the data in the cloud. You can move your data into the cloud, and also you can do analysis in the cloud. But why using the cloud for cancer research? And there are some advantages of using the cloud for cancer research. And here we are listing some of them. The first is that all the data on different types of data, for example, Fabian showed it to you, the different types of data that are available for cancer research. If you put all the data in the cloud, then they are in one same location. So you don't need to go into different web pages or different other computers to extract the data. So all the data is in one place. So that makes it easier to access and analyze the data. And not only that, we transformed the data into the same formats, and they are all in the cloud. So you don't need to pre-process most of the data that are available in ISB-CGC. They are already in the same format, so that you can easily process all the data that you want to use for your cancer research project. Also, for some of these analyses, typically you need to use a supercomputer. But if you don't have access to this supercomputer, then you can use one of the computers that Google Cloud or the cloud in general offers. And in this way, you can just use the amount of computing capacity that you need. So you can do the analysis in the cloud and optimize your costs. And the final point is that it's easy to share your results. You just need to give access to the cloud to your collaborators in order to share the data. So you don't need to transfer or send amounts of data through email or other ways. So those are some of the benefits that the cloud offers. And of course, it's not perfect for every project, but if you know the benefits, then you can take advantage of those and using the cloud for cancer research. And this is a snapshot of our webpage, so you can access it. We will be doing a tour over some of the resources later in this presentation. And now I will move a little bit into the data that we host in ISB-CGC. We host what is called derived data, and they are all in Google BigQuery. So they are all in a format that is very similar to an Excel table, so that you can easily access and process it. In the figure, you can see some of the data types that are in BigQuery tables. Sequencing data, methylation data, protein expression data, gene expression data. We also have clinical data available, microRNA sequencing data. And so we already process this data, so they are all in this table format that you can access it. And yeah, so one additional point is that it is very cheap to access this data. Even though I say this is publicly available, you still need to create an account and create a project in Google Cloud to be able to access this data. But all of this is publicly available. So anybody can access the data. And I will show you, I will explore the data a little bit later in this demonstration. And here is an example of a data type. And it is in a table, as you can see. I am pointing here an example of a preview of the data. So it has a columns, it has multiple properties in the different columns. And all the data that we host have this format. And in addition to the table, you also need to know about several properties, like the description for every column of the data, where it's located in Google Cloud, some description of the table, like the releases date and number of rows. All of this is available in the for every table. And we provide data from different projects, as Fabian showed it to you. Those same projects are stored in BigQuery tables in ISB-CGC. We have genomic data, proteomic data, we have reference information, we have clinical data, and it's easily also to move your data into the cloud. And if you are interested in a specific data set, then you can easily move your data so that you can combine your data with all other data from the different projects. And we can always help you also with that if you are interested in a specific data set. So there are two ways in which you can access the data. One is you have Google storage in which all the files are located. So you can also access the raw files from Google Cloud. And the second way is to use BigQuery, which is the set of tables that I just showed it to you. So those are the different ways in which you can access the data. And in the first, there are the raw files, and the second, there are transformed files into BigQuery tables. And this is the name of the project, ISB-CGC BQ, and I will show it to you later how to access it. Okay, now I will go over a web page, and I will show it to you some of the resources that we provide. I will be focusing on data access. We also provide other tools to process the data, but here I will focus mostly on how to access and how to explore all the data available in ISB-CGC. And the first resource that we're going to explore is the BigQuery table search, which will allow you to explore all the data that we host in ISB-CGC. So if you go to our web page, here is the link of our web page. If you go to our web page, then you will be able to access the BigQuery table search, clicking here. And it will take you to this interface in which you can search or you can explore all the tables. So the rows in this interface are the tables. And as you can see, there are many tables here. And it gives you information about which program this table belongs to, what category of data it has, if it is PDC or GDC, the data type, the number of rows, where it's created, and additional information. So for example, we can explore gene expression, for example, RNA seq. So it will search for all the tables that contains RNA-seq data. And we can specify additional filters. For example, we can select the program. And you can find here several programs. TCGA is the one that that is heavily used by cancer researchers. And we can find, for example, this table here. Although this is not RNA-seq. So we can use additional filters. For example, here, and we can find some tables. And this is the gene expression table that I was looking for. And if you click in this bottom, you will see additional information for this table, like the full name of the table, the data set it belongs, a little bit of description of the table, and for every column of the table, you have what this column is by having a little bit of a description and the number of columns. And you can even have a preview of the table, and this is the content of the table, and you can see the table a little bit. So this is a good resource if you are interested in a specific data type and you want to see if there are tables for that type. For example, we can explore if we are interested in clinical data. We can, I selected this. That's why it's giving me 0. But if you are interested in clinical data, for example, if you type here, it will provide all the tables that have or are related to clinical data. And again, you can select a program. I will select again TCGA, and it will filter TCGA related tables that have clinical data. So for every table, you have the information. In this case, there are many, many columns, and for each column, you have a description. And another aspect is that not only you can explore the tables, but you can also explore the data inside the table. And from this resource, if you click in here, which is this bottom, it will take you to an interface in which you can actually explore the data. So if you click in this bottom here, you will go to an interface in which you can process the data a little bit. So this is an interface provided by Google, and you have the same information here, but what I'm trying to say is that in addition to just looking the data, you have a preview, you can query the data, meaning you can filter the data. And so you can put some SQL commands here. So I prepare a small set of commands. So these are SQL commands, and it will filter the table that you are referencing. So this is the name of the table that we were exploring, and you are telling the interface to extract this column, which is the ID for every patient, and then you will extract the age for every patient and the vital status for every patient. And you specify the name of the table, and then you tell that you are interested only in cancer, in breast cancer. So this will select all breast cancer patients, and it will print the information about the IDs and the age and the vital status. So we click run, and it will give you all the list of cases for breast cancer, which are 1,000 cases for breast cancer, and it will give you also the age for every patient. Some patients doesn't have information for that for age and additional information. But the point is that this gives you an easy way in which you can do a little bit of statistics or filtering data that is available in ISB-CGC. And all started from this resource. So if you start from this resource, the BigQuery table search, you will be able to see the data, to explore the data, and do a little bit of analytics if you click into this bottom and go into the Google console, which is the interface that Google provides. So you need to know a little bit of SQL, but if you are doing simple tasks, then it is not very hard to do it in SQL. Yeah, so this is one of the resources that you can use for exploring all the data available in ISB-CGC. Going back to our presentation, we can go forward into the second. This is the console. This is a link so that you can access, you can go directly to this interface. So if you click in this link, you will go directly to the interface. But you can also use the Google query search, this interface, and click this button to go to this console interface. And the second resource is the cancer data file browser. So if we go to our webpage, and here is an interface in which you can select all the files available in ISB-CGC. So those are the raw files. For example, BAM files related to RNA-seq data, and also the files containing images that we also host. Those are pathology images, reports, radiology images. So all the files are here, and you can explore through this interface, all the files that are in ISB-CGC. This is an interesting resource, but as I mentioned before, we transform all these data into BigQuery tables. So now most of our analysis is done through BigQuery table. But but still, this gives you a way in which you can select all the files that are available in ISB-CGC, raw files. And the third resource is a data set. And this is, the Mittelman data set that store karyotypes, which contains chromosomal aberrations and gene fusion. So this is a very popular data set. It contains karyotypes which are aberrations in the chromosomes. For example, it contains, these aberrations can be deletions in chromosomes. For example, in this figure, there are some chromosomes that are missing. There are maybe some parts of the chromosome that is, for some reasons, deleted. Those are deletions that are common in cancer. There are also aberrations related to addition of unknown content or translocations when two chromosomes, for some reasons, combine it and transform the genetic content of a cell. So all these are chromosomal aberrations, and that are stored in this data set. This is kind of different from the genomic type, in that this is obtained from cells. whereas in the genomic, they use a sample. So it is the same kind of a similar, the similar data in the sense that both store chromosomal aberrations, but this tells you a little bit about for every cell, what is the aberration in the chromosome. So if you click here, you will be able to have an interface that will allow you to explore these chromosomal aberrations. And for example, we can select breast cancer. Things here. Breast cancer, and then we can select search, and it will give you a list of chromosomal aberrations. And this is the karyotype. This is the chromosomal aberration. And this has a specific format, but this is telling you that there is a chromosomal aberration, and this format will tell you a little bit about the details of this aberration, number of cases that are found in the data set, and importantly, it gives you the reference from which these aberrations was obtained. So for every case, it gives you the reference. And it is a list of all the aberrations that are found in breast cancer. So this is, there is a good guide, so you can look all the definitions of the chromosomal aberrations. And this data set was collected by Dr. Felix Mittelman, who started this data set, and it's very popular and is now in ISB-CGC, so that you can explore it, and also you can use BigQuery to explore this data set. And the final resource that I would like to show is the data set of TP53. TP53 encodes the protein P53, which is called tumor protein. Sometimes TP53 is also called tumor suppressor gene, and this protein is involved in cell division and keeping cells from growing and dividing. And it's very important in cancer research. I think it's one of the most studied genes in cancer research. And so that's why there are multiple data obtained from this gene. And there is available a database for this specific gene that contains different types of data. And if you go into our webpage and click in this resource, you will be going into this interface in which you can find the different types of data related to this gene, like functional or structural data, tumor variants, data from cell lines. These are not from patient samples, but from cell lines, data from mouse models, and other types of data. And we can explore the data a little bit. For example, if we want to explore gene variants, we can click into this, and then we will search for protein description, and it will give me a list of variants that happen in this gene. And it gives you additional information for each variant. Variants are changes in the gene. So this is a description of a variant, and it also gives you a reference from which this data is obtained. It has a good user manual, so you can read and explore all the data that is available for TP53. And you can also download the data. If you click download, you will be able to get all this data. And It's taking some time, but it's another way to explore the data. So in summary, this is another TP50, another resource that we provide, and it has all the data available of the TP53 database. So those were some of the resources that we provide. So a BigQuery table search that allows you to explore all the data that is available in BigQuery tables that are hosted in ISB-CGC. The cancer data file browser that allows you to explore the files, the original files that we store in Google Cloud. This data set, the Mittelman database of chromosomal aberrations, and also recently the TP53 database that has many different data types for TP53, which is a very important gene in cancer research. Finally, all this data can be accessed through not only through the resources that I showed to you, but also you can use R and Python to access and explore the data. And this is very important because when you are doing a project and writing a paper, you need to do some statistical analysis of your data, and typically R or Python are used to do this analysis because they provide different packages. And also they have tools to generate figures so that you can create figures that are ready for your paper using these resources. We provide notebooks that show it to you how to use R and Python to explore and analyze all the data that we provide. And you can go into this link to access all these notebooks. And if you are interested, then we can have another session in which we can show how to use R or Python to access and perform statistical analysis of ISB-CGC data in the cloud. And of course, if you have additional questions, you can always send us an email. This is an email that we provide. And if you have specific technical questions, you can also go to our office hours. That here are some information about office hours 2 times a week. So we will be happy to help you and answer your questions. And that's pretty much it. Thank you very much for your attention. And if you have questions, please let us know.
Thank you very much, Dr. Aguilar, and Fabian and Poojitha. I think this has been a very exciting overview of the resources that are available. I'm sure everyone here will agree that there's a lot of data here, and this gives us an opportunity to work, to mine data, to go from data to information, essentially. Are there any questions from the audience? You can indicate it in the Q&A or in the chat, or we can give you access to voice your question if you so indicate, if you so prefer. Anybody from the audience?
So Boris, I had a question. You touched on some of the notebooks that we have in ISB-CGC. Can you talk a little bit more about what kinds of analysis researchers can do with the available notebooks with the data that we have?
Yes, I think one basic type of analysis is getting some statistics from the patients, for example. And then there are different use cases that we provide for using genomic data. For example, identifying, if you compare normal samples and cancer samples, and you want to identify the differences in genes expression between these two samples, then you can use this data, and we provide some notebooks for that. And for the different types of data, there are use cases available for example, for mutations. If you want to know the frequency of mutation for breast cancer patients, then you can use the notebooks that we provide as well. So it depends on the data that you have. You can identify different use cases, and some use cases are available in ISB-CGC notebooks.
Yep, awesome. And I see the link has been posted there. It is in the slide as well. So if anybody's interested to explore those community notebooks, they can just go to that link, and it is a public link, if I'm not wrong, right, Boris, that anybody can access?
Yeah.
If you can put it in the chat, that'd be great. Yeah.
Yeah.
I will do it. So we have Q&A, but I don't see a chat here.
Oh,
I think you might need to
chat. Okay.
ding.
I will put for
I can try to put it, since I can see the chat on my end, so I'll paste the link here.
Oh, thank you.
yeah. I know that there are some individuals from the University of Ghana who have a lot of background in programming, and I think perhaps as a follow-up at some point we can have we can do a project and then we can get some help.
Yeah. So
with walkthrough.
If we go to this link, hopefully you are seeing my screen.
Yeah, you can see your screen.
So we go to the link, you will find a GitHub repository, and in every folder you will see different notebooks. Okay. But for example, here there's a teaching material that we collected for another session, another talk, and here is where you can find some of the notebooks that provides like introduction to BigQuery and how to analyze the data, and examples of statistical analysis, like for example, survival analysis, and also machine learning. There are some examples with machine learning there. So yeah, so you can explore this link. There are many notebooks there.
Okay. There's a question in the Q&A. Boris, or Fabian, or Poojitha.
Can you
take that one? The question is, can you elaborate on the data sources and how this could be useful, particularly to African researchers?
Yeah, so the data sources are mainly these Western American initiatives. So there's limited data available for African patients, I'm pretty sure. I feel like the best use case would be to sort of use as a comparison, as a teaching tool, just to learn the methodologies. And I went to AACR last week, and there was a lot of GWAS studies, for example, that looked at the ability of European data sets to predict cancer mutation, predictive power in South American populations. And it seemed like there was a substantial amount of overlap in predictive power of some of those mutations. So just using it as a tool to sort of enrich what might be smaller data sets that are available from African researchers. We also have, if you're running an analysis in the cloud, we have $300 worth of free cloud credits. So those are, that's funds you can use to run whatever project you're interested in the cloud and using cloud resources. It doesn't necessarily have to use GDC data. So that's also, hopefully that's addressing the question.
Thank you very much.
And of course, I feel like the Google Cloud is also a fantastic tool to use for your own data. It's centralized. I think they have African servers as well. And the real benefit is that it's really, really easy to share this, your data with other researchers. So say you generate an RNA sequencing data set in your lab, you can toss it into a BigQuery table, give permissions to all the members of your lab. And then if there's ever any follow-up question that wants to reuse the same data, it's no longer, oh, hey, can you send me that, text file that you have on your computer to me? And then, there's multiple different versions and there's confusion about which one's the most recent. You can just kind of direct them to the BigQuery table. And it's, it isn't stored on one person's computer, and it kind of bypasses some of those data management issues that crop up very frequently, I feel like, in research labs.
Yep, and I just want to point out that if any one of you does look at the notebooks and the data sources and tries to either incorporate it in your own pipelines or use it in your research, just want to point out that we do have office hours again. Thank you, Boris, for putting that on the slide. So we have bioinformaticians in our team that can answer your questions when you do come across questions with data, especially data from any of the data commons. And if you join the office hours, one of us will be able to either answer your question or point you to the correct resource that can help you.
That's very helpful. Feedback at ISB-CGC.org. Yeah. All right. Thank you very much. This has been a wonderful overview of the resources and the tools available. We will do a survey and get some feedback as to specific interest groups on some of our campuses and discuss follow-up sessions from there. So let me take this opportunity to thank you all, Fabian, Boris, Poojitha, and thank you all for your time. I know it must have taken a good deal of time putting this together and sharing it, as well as all our participants.
Thank you for having us. Thank you for giving us the
Thank you for
opportunity
your time.
to
Thanks,
share our
George.
resources.
Thank you.
Thank you, guys. Bye.
Thank you.